Let's start with the introduction of Williams syndrome, also known as Williams-Billings syndrome, a developmental disorder which is multi-system disorder affecting many parts of the body. And it has incidence of 1 in 7,500 to 18,000 individuals and can equally affect both males and females. It is a chromosome disorder where there is 1.5 MB deletion of proximal long arm, that is Q arm of chromosome 7, which contains almost like 30 different protein coding genes. For example, elastin gene. Most cases of Williams syndrome are not inherited and usually occurs as a random event. However, small percent of the cases are due to autosomal dominant inheritance pattern, where patient inherit the chromosomal deletion from a parent who has the condition. And people who are suffering from Williams syndrome has life expectancy lesser than general population. Before we talk more about Williams syndrome, I'd like to discuss about chromosomal rearrangements, which can be divided into four types: duplication, deletion, inversion, and translocation. In case of duplication, a part of chromosome gets doubled, and it can be tandem or dispersed duplication. They can be on the same or reversed order of the original. Unbalanced duplications affect gene expressions. Talking about deletion, which we can see in Williams syndrome, it is a loss of chromosome segment which can affect several genes. Deletions can cause unbalanced gene expression in a region, and the position inside the deletion has big impact on phenotypic consequences. Inversion is when a segment of chromosome is inverted or flipped, and there are two types of inversion, which are paracentric inversion, which do not include centromere, results in huge deletion, and pericentric inversion, includes centromere. They exchange information unevenly, results in duplication and deletion in both chromatids. A translocation is movement of a part of one chromosome to non-homologous chromosome, and they can be balanced or unbalanced. Talking about balanced rearrangements have no loss or duplication of genetic information. Hence, individuals who have balanced rearrangements are phenotypically normal. And unbalanced rearrangements occurs when there is some or loss of duplication of genetic material in an individual leading to phenotypic changes. Williams syndrome is caused by pathological loss of Williams syndrome critical region WSCR or 1.55 to 1.83 MB region on chromosome 7. The WSCR frequently undergoes rearrangement due to the presence of large complex segmental duplication termed low copy repeat LCR. The low copy repeats mediate non-allelic homologous combination between the highly similar DNA sequences during the meiosis, resulting in an increased rate of de novo copy number variation within the region. If the deletion comprises smaller segment of the WSCR in an individual, it shows lesser phenotypic feature on them, and if the deletion is larger which extend beyond the WSCR, it causes additional phenotypic features. Williams syndrome can cause various symptoms that range in severity. Since it's a multi-system disorder, there are various characteristic symptoms which include developmental delay, intellectual disability, hypotonia as a neurologic symptoms. Distinctive facial features like small jaw, large ears, epicanthal fold, blue eyes, wide mouth, etc. There are various behavioral symptoms which includes anxiety over friendliness with stranger and ADHD. Musculoskeletal disorders include scoliosis, short stature, and most importantly, cardiovascular disorders which include supraventricular aortic stenosis, hypercalcemia. In this slide, We have pictures of symptoms that occur in Williams syndrome, a form of cardiovascular disease called supraventricular aortic stenosis occurs frequently in people with Williams syndrome. Supraventricular aortic stenosis is a narrowing of the large blood vessel that carries blood from heart to the rest of the body. And if this condition is not treated, the aortic narrowing can lead to shortness of breath, chest pain, heart failure, and death may occur. The deletion of loci within Williams syndrome critical region (WSCR) that we discussed in previous slide include the elastin gene, which is responsible for the cardiovascular condition here, supraventricular aortic stenosis. We know that the proximal long arm of chromosome 7, 7q11.23, contains almost 30 genes and several non-coding RNAs. Knowledge about how each of these genes contribute to the Williams syndrome phenotype is still growing. Elastin gene is one of the genes present in loci of chromosome 7 which causes Williams syndrome. Function of elastin gene is to encode a protein which is one of the two components of elastin fibers. Elastin fibers contain part of extracellular matrix which gives elasticity to organs like lungs, skin, blood vessels. An encoded protein is rich in glycine and proline, and cross-linking with lysine residue which takes place extracellularly gives elastin its elastic properties. And elastin is broken down by elastase. Oh, it's really confusing, like a tongue twister. Deletion or mutation within the loci which has elastin gene is associated with cardiovascular manifestations such as stenosis, which can globally narrow and thicken blood vessels. Elastin insufficiency can cause connective tissue disorders like premature aging of skin due to decrease in dermal collagen and elastin. The condition is typically diagnosed in early childhood. If your healthcare provider suspects your child has Williams syndrome, they'll provide a physical examination of your child, which is followed by genetic test, which is a blood test that identifies gene differences, and the most widely used method is FISH. Fluorescence in situ hybridization is a type of specialized chromosomal analysis where a blood sample is taken and chromosome 7 is treated with two specific colored markers. Then it detects the absence or presence of elastin gene. If the person has only one copy of elastin gene, then the diagnosis of Williams syndrome is confirmed. Here in the picture, we have FISH analysis of Williams syndrome with positive Williams syndrome on left, where elastin gene is found only on one chromosome. The other copy of the gene is deleted, hence the patient has Williams syndrome. And on the right side, we have FISH analysis of negative Williams syndrome, where elastin gene is found on both chromosomes. Hence, the individual does not have Williams syndrome. In this slide, we talk about advantages and disadvantages of FISH. Advantages of FISH is they are readily available at major hospital and cytogenetics lab. They have high sensitivity and may detect translocation. Disadvantages of FISH is they are higher cost, coverage of insurance varies, they cannot detect microdeletion, hence there is higher chance of false negative results. And it is not a routine test, therefore labs need to be called in advance to order correct probe. Summarizing the disease, we know that Williams syndrome is a genetic disorder where there is deletion of chromosome 7q11.23, which can be identified by its distinctive features like intellectual disability, small jaws, short statures, etc., and the diagnosis is confirmed by FISH analysis. Individuals who do not have gene deletion do not have the disease. Prenatal screening is non-specific, where ultrasound can detect intrauterine growth retardation and various cardiovascular anomalies. Management of the disease is symptomatic treatment, regular cardiovascular monitoring, physiotherapy, medical treatment for hypercalcemia, surgical treatment for any cardiovascular issues, support and educating the family.